Imagine living 10,000 years ago. You are standing in a dense, primeval forest. Someone in your tribe suffers a deep, feverish wound. Their skin is hot, their body is shaking, and pain radiates through their limbs. There are no hospitals, there are no pharmacies, there are no microscopes to reveal the invisible bacteria invading their bloodstream. All you have is the earth beneath your feet, the plants growing around you, and thousands of years of trial and error. How did early humans figure out that chewing the bark of a specific willow tree could dull a blinding headache? How did they know that certain molds could heal an infection, or that a single drop of plant sap could either save a life or end it instantly? For most of human history, medicine was not a science. It was a high-stakes game of observation, intuition, and survival. It was a bridge between nature, magic, and accidental discovery. Fast forward to today. Today, a scientist sits in a climate-controlled laboratory surrounded by automated supercomputers. They don't search through forests for remedies. They design molecules on a screen, atom by atom, targeting specific protein structures inside a single human cell. How did we bridge that vast gap? How did humanity transform raw, chaotic nature into modern, precision pharmacology? Over the next 20 minutes, we are going to embark on an incredible journey through time. We will explore how ancient civilizations discovered the hidden chemistry of plants, how early pioneers isolated the very first miracle drugs, and how modern science uses computational chemistry to manufacture molecules that nature never even dreamed of. Welcome to the story of how we learned to cure disease. Long before humans wrote books about medicine, we learned by watching the natural world. If you observe animals in the wild, you will notice something extraordinary. When bears emerge from hibernation with parasites in their stomachs, they actively seek out specific bitter roots that act as natural dewormers. When wild chimpanzees feel ill, they swallow whole, rough leaves that scrape parasites from their intestinal tracts. This behavior is called zoopharmacognosy, animals using plants and minerals as self-medication. Early humans were master observers. They watched which plants animals consumed when injured or sick, and they began experimenting on themselves. Of course, this early experimentation came at a deadly cost. Without understanding chemical dosages, the line between medicine and deadly poison was razor thin. If a tribal healer gave a patient too little of a toxic root, it did nothing. If they gave too much, the patient died. Through generations of oral tradition, ancient cultures built massive mental libraries of botanical knowledge. In ancient Sumeria, 5,000 years ago, clay tablets recorded the medicinal uses of opium, poppies, myrrh, and thyme. In ancient Egypt, the famous Ebers Papyrus, written around 1500 BC, detailed over 800 medical remedies. The Egyptians used honey to treat open wounds, unaware that honey naturally produces hydrogen peroxide and creates an antibacterial barrier. They applied moldy bread to infections, unknowingly using the primitive precursor to penicillin over 3,000 years before Alexander Fleming. Meanwhile, in ancient China, the legendary Emperor Shennong is said to have tested hundreds of herbs on himself. Chinese medicine identified sweet wormwood as a cure for fevers. Thousands of years later, modern scientists would isolate the active compound from that exact plant, artemisinin, which became a Nobel Prize-winning treatment for malaria. And in ancient Greece, the bark of the white willow tree was crushed to treat pain and reduce fever. The ancient world did not understand why these remedies worked. They attributed cures to gods, spirits, or the balance of bodily fluids, but their empirical observations were dead right. Nature had spent millions of years conducting its own chemical experiments, and humans were finally learning to read the code. For thousands of years, medicine remained crude. If you needed pain relief, you drank a bitter tea brewed from plants. The problem with herbal brews was unpredictability. The potency of a plant depended on the weather, the soil, the season, and how the herb was prepared. One cup of willow bark tea might do nothing, while the next cup might cause severe stomach bleeding. The true revolution in medicine occurred in the early 19th century, when science learned to stop using the whole plant and start isolating the active molecule. In 1804, a young German pharmacy apprentice named Friedrich Serturner was experimenting with raw opium poppies. He succeeded in doing something no human had ever done before. He isolated a pure, crystalline chemical compound from the plant fluid. He named it morphine, after Morpheus, the Greek god of dreams. This was the birth of modern pharmacology. Scientists suddenly realized that plants weren't magical. They were simply biological factories manufacturing tiny, specific chemical keys, and those chemical keys could fit into the biological locks inside the human body. A few decades later, chemists turned their attention back to the ancient Greek remedy, willow bark. Scientists isolated the active ingredient, a compound called salicin. But raw salicin caused severe stomach irritation. So in 1897, a German chemist named Felix Hoffmann modified the molecule by adding an acetyl group, creating acetylsalicylic acid. We know it today as aspirin. Aspirin became the first mass-produced synthetic drug in human history. It proved that humanity didn't just have to accept what nature provided. We could take nature's molecules, refine them, and make them safer and more effective. Then came the accidental miracle that changed history forever, the discovery of penicillin in 1928. Alexander Fleming returned to his messy laboratory after a vacation and noticed that a stray mold called Penicillium notatum had contaminated one of his petri dishes. Around the mold, all the deadly Staphylococcus bacteria had dissolved. The mold was secreting a chemical weapon to kill off competing bacteria. For the first time, humanity had a weapon against bacterial infections. Simple scratches, childbirth fever, and minor wounds were no longer automatic death sentences. We had transitioned from picking herbs in the dirt to extracting life-saving chemical weapons from microscopic fungi. Today, if you take a prescription medication, chances are zero that a scientist went out into a forest to harvest it. Modern medicine is manufactured through advanced synthetic chemistry and biotechnology. So, how do modern pharmaceutical companies build a drug from scratch today? It begins not with a plant, but with a target. When medical researchers want to cure a disease, whether it's high blood pressure, diabetes, or cancer, they first study the human body at the molecular level. They identify a specific protein, receptor, or enzyme that is malfunctioning. Think of this malfunctioning protein as a broken lock on a door. Once scientists identify the target, the search begins for a molecule that can act as the perfect key. In modern drug discovery, scientists use a process called high-throughput screening. Automated robotic arms test hundreds of thousands of synthetic chemical compounds against the target protein in real time. If a molecule binds to the target and stops the disease process, it becomes a lead compound. From there, medicinal chemists take that lead compound into the lab. They tweak its atomic structure, adding a carbon atom here, removing a hydrogen atom there, optimizing the molecule so it hits the target precisely without causing toxic side effects elsewhere in the body. Once a safe, effective molecule is engineered, it moves through rigorous testing, in vitro testing, testing the drug on human cell cultures in petri dishes. Preclinical testing, evaluating safety and metabolism in animal models. Phase 1. Clinical trials, testing small doses on healthy human volunteers for safety. Phase 2 and 3. Clinical trials, testing on thousands of patients to prove efficacy against placebo. Out of tens of thousands of candidate molecules created in a lab, only a tiny fraction ever make it to your local pharmacy shelf. It takes over a decade and billions of dollars to bring a single modern drug to market. It is one of the most complex, rigorous scientific processes in human history. As impressive as synthetic chemistry is, we are currently standing on the threshold of an even greater medical revolution. For the past century, most drugs were small molecule chemical drugs, simple compounds like aspirin or ibuprofen built in chemical reactors. Now we have entered the age of biologics and mRNA technology. Instead of using simple synthetic chemicals, biologics are complex, giant proteins manufactured inside living cells. We are talking about monoclonal antibodies engineered to hunt down specific cancer cells like microscopic heat-seeking missiles, leaving healthy tissue completely untouched. Even more revolutionary is the shift from giving medicine to instructing the body to make its own medicine. Consider mRNA technology. Traditional vaccines introduce a weakened or inactivated virus into your body so your immune system can learn to recognize it. mRNA vaccines don't contain a virus at all. Instead, they deliver a tiny strand of genetic code, a set of biological software instructions. Your own cells read the code, temporarily produce a harmless viral protein, and train your immune system to defend against the real threat. And now, artificial intelligence is accelerating this entire process at breakneck speed. In the past, discovering a new drug candidate took years of physical laboratory trial and error. Today, AI models can simulate the 3D structures of millions of proteins in seconds. They can predict exactly how a synthetic molecule will fold and bind to a human receptor before a scientist ever mixes a single chemical in a beaker. What used to take a decade of human effort can now be modeled in a matter of days. We are moving toward an era of personalized medicine, where treatments will be custom synthesized based on your unique genetic profile, eliminating side effects and maximizing cures. When you look back across the vast expanse of human history, our struggle against disease is a testament to human resilience. We started in the dark, chewing bitter leaves, rubbing clay on wounds, and hoping the gods would smile upon us. We suffered immeasurable loss, but through endless curiosity and observation, we slowly unraveled the chemical secrets of the natural world. We transformed wild willow bark into aspirin. We turned stray mold into penicillin. And today, we write genetic code to train our own immune systems to fight global pandemics and incurable diseases. Yet, nature is always evolving. Bacteria adapt to our antibiotics, viruses mutate, and new health challenges emerge with every generation. The story of medicine is not a finished book. It is an ongoing battle between human ingenuity and biological evolution. The next time you take a simple pill for a fever or an infection, take a brief moment to reflect on what is sitting in the palm of your hand. You aren't just holding a tablet. You are holding thousands of years of human trial, sacrifice, discovery, and scientific triumph. The quest to heal is as old as humanity itself, and the greatest breakthroughs are still ahead of us. Thank you for watching.